Hey bio class, I hope you guys are enjoying the beautiful weather we've had the past couple days. I certainly am. As you can see, I am outside somewhere. If you can figure out where, put it into my email, send me a message, say, hey, I know exactly where you are, and tell me, and maybe I'll get you a few bonus marks on your next assignment in genetics, because that's the first this is the first lesson in this new unit on genetics. So we have now finished the plant unit. So when we are looking at genetics, what are we looking at? Well, we have this specific molecule, and the molecule is called deoxyribonucleic acid, which is kind of a mouthful, so we don't say that. We just say DNA. Okay, so DNA, we have figured out, is responsible for carrying traits. So when we talk about the different traits, what are we talking about? Well, these are things that we see in us or in whatever we're looking at that are measurable. And in the case of some traits, they come from your parents through your nurture, being brought up that way, but others are genetically programmed into you. Now, some are argued back and forth as to whether you're born this way or, you learn these things. Some people used to talk about having the double Y chromosomes as making people super males, and so they were more aggressive. And so people tried to argue when they happened to have that and they were a criminal of some kind, and they said, well, I'm predisposed to it, I can't help it, and so you can't, hold it against me that I'm programmed to be this way. And of course that's been shown to be not true. Super males, yes, you have the double Y, but it doesn't make you any more aggressive or anything like that. And no, it doesn't turn you into some kind of superhero. So things like that. So when we look at that kind of stuff, those are subjective. So we're going to focus on traits that, without a doubt, those are from your genes. For example, my eye color, your hair color, okay, things like that. All of that is programmed into your genetics, and that's just the way you are. How short I am. I know I'm, average height for a woman, I think. So I'm a little short, but I can thank my parents for that because they're both short. And interestingly enough, when you look at traits, shortness is actually a dominant trait in humans. So if you have two tall parents, their kids are going to be tall no matter what. But if you have short parents, there's still a chance that the kids are going to be tall. So I'm really hoping my kids can get some height because my wife's tall, and maybe I just might have some height in my genes somewhere. Anyways, moving on. Looking at DNA. Well, what is it? Well, it's an organic molecule, and when you get into chemistry next year, you learn a little more about organic chemistry. And also, if you take the 4U bio next year, you also learn a little more about organic chemistry. And organic molecules are just carbon-based molecules that are kind of building blocks of life. Well, the DNA is a organic carbon-based molecule. And what is it made up of? Well, it has a sugar, which is the ribose, and then it has a phosphate backbone, and then we have nitrogenous bases. So here's kind of what your molecule, one little part of your DNA. Okay, this is your nucleotide base, and so what we have here is the phosphate group, the sugar, and then there's the nitrogenous base right here. Okay, and there are 4 main types in your DNA. We have adenine, thymine, guanine, and cytosine. And notice I put these little connectors here. That's telling you adenine always binds with thymine, and guanine always bonds with cytosine. And they link together not through a chemical reaction, but actually through hydrogen bonding. So it weaves itself together in these double-stranded helix. So you have these base pairs. So if this was adenine, that's thymine. If this is cytosine, that's guanine. And the reason it's not chemically linked together with a chemical bond, but a hydrogen bond, is because the bond's strong enough to hold if you have a bunch of them together. Think of water sticking to your finger. It's a hydrogen bond. It's attracting itself. Oh, I just dropped my mouse. Okay, it's attracting itself to your finger. Well, the same thing's happening here, but it doesn't require very much energy to separate. So DNA is constantly being zipped and unzipped to be read and produce different proteins and all this within your body to keep the cell going. But if it required a chemical reaction to actually break the DNA apart, it would be impossible for life to exist. It would be too much energy. We just couldn't do it. Think about a single strand of DNA being 6 feet long, and that's in every single one of your trillions of cells in your body. So if you required that amount of energy, it just couldn't happen. So we need these hydrogen bonds that pull everything together. Okay. Now down at the bottom, this is a weird one, uracil. Now uracil actually replaces thymine as the part of the sequence, and it bonds with adenine in that case. So when you're making RNA, and you learn about this in grade 12, there's different types of RNA that send out messages in your body. Well, instead of having thymine, it's going to have this uracil stuff. So down here, here's the bases. So we have adenine and guanine, and see they have kind of similar structures. There's just different attachments to them. So you can see here there's an ammonia and an oxygen versus the ammonia over here, and there's nothing down here, and then there's an extra H attached to that nitrogen right there. No double bond. Okay, so just a few little differences, and then down here, here's cytosine and thymine, and also uracil. So you can see how uracil is so similar to thymine, and so that's why it replaces the thymine in the RNA. And there are some things out there that are RNA based. That's a lot of viruses are RNA based instead of DNA based. Now DNA, here's the whole packaging thing of DNA. So as I said, when it's stretched out, it's about 6 feet, 2 meters approximately in length. If you just took the DNA and stretched it out in one single cell of the human, that's crazy. But what it ends up happening is the DNA is wrapped together and wrapped again, and then it goes around these little histones, which are a type of protein molecule which protect it, and then those then wrap and coil some more and some more and some more until you finally get into the chromosome. And when the only time you can actually see the DNA within a cell when you're looking under a microscope is when it has shortened and thickened in the process of mitosis. Sorry, my leg is getting tired sitting here. Anyways, so yeah, once things are going through mitosis, and you've seen this in the onion root tip cells and things like that when I made you do diagrams, you see the sister chromatids paired up and they're shortened and thickened. Okay, moving on. So again, DNA, where is it? It's found in most cells, okay, and the reason I say most types of cells, well, our red blood cells don't actually have DNA because there's no nucleus, and prokaryotes, they have DNA, but they don't have a nucleus. So when you look at a prokaryote, they just have the DNA kind of hanging out in the middle of the cell somewhere. So what do we actually know about cells? Let's look at that for a little bit. So the first part, as a review, Robert Hooke, the first guy to coin the term cell, and he saw that when he looked under his microscope, and that's his homemade microscope right there, and this is what he saw. And he looked at that and said, hey, those look like jail cells, and so he called them cells. Okay? Now, the next thing, scientists used to think way back in the day, because when they left food out or things like that, you would see animals start growing, or fuzz or whatever, growing on the food and rotting it. And so they thought that life spontaneously generated out of nowhere and just kind of grew from nothing and turned into life. Well, we had this guy, Louis Pasteur, who went out there and said, no, that's not true at all. I'm going to show you that life just doesn't come from nowhere. It has to come from something. And so Louis Pasteur went out and performed this famous experiment, and I'm going to show you here. Basically, he filled this flask with a type of broth, and he would heat it up and turn it into a swan-necked flask. So the glass is stretched out like this. And then he would leave this end open, and he would heat the whole thing, sterilizing it. And once he heated it and sterilized it, he would seal these ends. And then he would go around all over the place, and he would break, he left a few just completely sealed, and other ones he would just break the tip here and just leave them. And then other ones he would tip, like you can see here, and then tip back. Now what he noticed was the ones that were sealed up, nothing grew, but he also noticed that the ones that weren't tipped over, nothing got into it, and it didn't spoil, it didn't go bad, it stayed perfectly fine. And then the one that he tipped ended up growing things. So he's showing that unless something got in there to reproduce, nothing is going to happen. And so life still has not generated in these swan neck flasks. They've stored them in a few different museums around the world of his flasks from, you know, about 200 years ago, something like that, and they still have not spoiled, the ones that he, you know, even opened the end to. They still have not rotted or gone bad. So just showing you that. And that led to the cell theory. Now the cell theory, what does it say? Well, the first part, all living organisms are made up of cells, whether it's one-celled organism or multi-celled organisms. Obviously the simplest unit of life is a cell, so there's been a lot of argument out there as to whether viruses are alive or not, and it's, you know, something that applies to nowadays because we've got the coronavirus, right? Well, technically, viruses can't survive on their own, they can't reproduce on their own, they can't even feed themselves. So as a result, viruses, for the most part, most people will conclude they are not alive. They need a host, and they don't just need the host, they need all the nutrients, and they need everything to build their genetic information so it can be passed on. So as a result, they're not considered alive. So the smallest units of life really are the bacteria that we see out there. And then finally, the last one, and this is thanks to Louis Pasteur, all cells come from pre-existing cells. So cells don't just form life. They can't just come together and make all these complex processes happen, because every process that happens in a cell had to be there at the beginning for the cell to function. If that doesn't happen, well, the cell doesn't live. Okay? Okay, let's move on. So of course, we know that there are all these other different functions within a cell, and there's all these different parts. We have all the organelles that are present within cells. But of course, what are we looking at? We're looking at DNA. And DNA is the thing that is controlling the functioning of the cell, making sure it does what it's supposed to. And of course, in order for cells to survive and for multicellular creatures to be multicellular, you have to replicate all of that information. And so we are looking at cell division and mitosis and kind of how that is going to work. So I am going to stop here, and that'll be a lesson for another day, looking at the process of cell division. So you guys have a good one, and hopefully you enjoy the weekend. See you around.